Hi and good day to everyone. In this video, my group will discuss about cystic fibrosis. These are my group members, so these are the elements that we will talk about. For the first part, which is introduction, cystic fibrosis or CF is a genetic disease that affects the lungs, digestive system and other organs. This happens due to the protein that creates and produces mucus in every organ doesn't function and not working properly. It causes thick and sticky mucus to form which can cause damage to the organ. The symptoms are vary from person to person. The possible symptoms are shortness of breath, abdominal pain, diarrhea, nasal polyps, jaundice, and limited growth or weight gain in kids. CF affects around 30,000 persons globally, which is more than 70,000 worldwide. From the statistic, by the age of 2, more than 75% of people with CF have been diagnosed, while 25% of the CF population is over the age of 18. However, CF is still an uncommon condition in Malaysia.
Next, we shall talk about the CFTR genes. CFTR, or the cystic fibrosis transmembrane conductance regulator genes, are genes which transcribe for the CFTR protein. It is located on the long arm of the chromosome 7 at position 31.2, and it contains 27 exons. The CFTR proteins are used as channels across membrane cells that produce bodily fluids such as mucus, saliva, sweat, etc. This channel controls the movement of chloride ions in and out of the cells. The transport of the chloride ions aid in regulating tissue water flow to ensure the creation of thin, free-flowing mucus. Mutations to this gene causes a disease known as cystic fibrosis. Cystic fibrosis is an inherited disease characterized by the buildup of thick, sticky mucus that can damage many of the body's organs. The thick mucus is due to the dysfunctional protein channel, causing an improper regulation of water for mucus production. Patients would notice respiratory problems, thick mucus production, and chronic lung illnesses.
There are two treatment options for cystic fibrosis involving gene therapy and gene editing. Gene therapy is the most common and conventional treatment for this disease. It can be done via inhalation or in vitro by replacing the defect CFTR gene with the correct one. In vivo gene therapy, specialized therapeutic vector is used to take a new normal CFTR gene and deliver into individual cells to make functional CFTR protein. There are types of vector used including viral vectors such as adenoviral vector and lentiviral vector, or non-viral vectors such as exosome, liposome, and lipid nanoparticle. The therapeutic vector is then delivered to the epithelial cell in patient airways.
Biotechnology can be used to cut out and replace defective part of CFTR protein in DNA, such as reverse Cas9. It has three significant components, which are the repair template, guide RNA, and Cas9 nucleases. Once these three components are introduced into the cell, the guide RNA binds to and express the Cas9 nucleus. The molecular scissors can search the cell's DNA for potential targets at the potential target site. The Cas9 unwinds a section of DNA to see if the guide RNA sequence matches. It then cuts the DNA to create a break in the strand. To replace the mutation with a normal sequence, repair process uses the precisely edited DNA. For this editing process to work, a short piece of DNA that contains the desired typical normal gene sequence as a template is introduced into the cell. The cell uses the corrected DNA strand to produce a functional CFTR protein that allows the proper flow of salt and fluids through the cell.
In conclusion, gene therapy is a way of replacing faulty genes with normal ones, as it is a method of managing cystic fibrosis. Genome editing is at the forefront not just by offering novel therapeutic solutions, but also for the development of experimental model, cellular and animal, which is essential in translational medicine. This method promising outstanding results, suggesting that cystic fibrosis will become a thing of the past.